In grand rounds. Dr. Deborah Topmeyer is here. She's going to introduce our speaker for today.
Absolutely. It is my pleasure to introduce my friend and colleague, Dr. Shridar Ganesan. Shridar is a medical oncologist and physician scientist at Rutgers Cancer Institute of New Jersey. And he's truly a Jersey boy, having grown up in East Windsor. He obtained his AB in chemistry from Princeton University and then received his MD and PhD in cell biology at Yale University. Working up the coast, the academic coast, he completed his residency training in internal medicine at the Brigham and Women's Hospital, where he also served as chief resident. He then completed a fellowship in medical oncology at the Dana-Farber Cancer Institute and further postdoctoral training prior to being recruited at CINJ in the laboratory of Dr. David Livingston. He was recruited to CINJ more than 10 years ago as an assistant professor, and we were really delighted to get him and are very thankful to Dr. Arnold Rapson. And he established his laboratory focused on DNA repair and cancer genomics. He has made significant contributions to the understanding of the role of BRCA1 in DNA double-strand break repair, the interplay between genetic and epigenetic stability, and the regulation of DNA repair choice. More recently, his laboratory has focused on understanding mechanisms of sensitivity and resistance to both targeted therapy and immunotherapy of cancer. His work, quite impressively, has been continuously funded by the NCI for the past 15 years and has led to many high-impact publications. He is now a professor of medicine and pharmacology. He holds the Omar Boraie Chair in Genomic Science, serves as the chief of the section of molecular oncology and associate director of translational research at Rutgers Cancer Institute, and leads the precision oncology effort at the Cancer Center. And I must say it was one of the earliest programs in the country. I will say, despite all these achievements, what is most impressive about Shridar is his remarkable heart and soul of collegiality and mentorship. And that has led to multiple junior faculty being awarded their first R01s. And Shridar truly defines the triple threat academician, outstanding achievement in the triad of research, education, and service. And without further ado, Shridar.
That's a very kind introduction. I'm blushing and I need my mother to hear it. I really appreciate the opportunity to talk here today. And let me share the screen. And you know, I'm a medical oncologist. I kind of focus on breast cancer and rare cancers, and I have a laboratory that's really focused on fundamental aspects of DNA repair. But today I'm going to talk to you some work we've been doing, really looking at genomic sequencing in the clinic. All right. And when we talk about kind of a molecular analysis of cancer, the thing I always start when speaking with trainees is kind of a, stepping back. These are my disclosures. So yeah, I do consulting for both big pharma and small pharma, and I'm on the scientific advisory board of a couple of startups that spun out of both Rutgers and Princeton. And my wife is an employee of Merck. So one of the things we always talk, before we kind of get into the molecular details, I always like to step back and reassess, a fundamental question is, what is cancer? How do we define the set of diseases that we kind of put under the rubric of cancer? And when I kind of ask trainees this, and this is an honest question, all right? When I ask trainees this, and back in the days when we used to actually talk in person, I'd ask trainees in the audience what they think are the hallmarks of diseases that are classified as cancer as opposed to another kind of disease. And people say, it's a disease of uncontrolled proliferation that makes you sick. But actually, you can think of other examples, like, for example, hypothyroidism. There you can get, due to autoimmune stimulation, enormous growth of the thyroid, and that can make you quite sick. But we don't consider that cancer. People say, what about if disease that grows and metastasizes, spreads to other parts of the body and makes you sick? Well, you can think about, what about endometriosis? There you literally get metastasis of the endometrial lining into the peritoneal cavity. And that also can make people quite ill, but somehow we don't consider that cancer, even though that might be changing in a little while. All right. And so the idea is that actually it's very hard to describe how the set of diseases we define as cancer is distinct. And you can think about it as characterized by unrelenting, disorganized growth of cells or tissue that doesn't respect tissue boundaries, and importantly, left alone will make you sick. All right. So this is a functional definition. And it's kind of like how we define infection. So this is really saying that cancer is not a disease, but describes a pathological and clinical state that's comprised of many, possibly thousands, of distinct disease entities with separate pathophysiology, molecular etiology, and biological behavior. It's very similar to saying infection, which also is very hard to define clinically and made up of many disparate entities. And put it in perspective, we never say, what's the, when are we gonna get the cure for infection, or what's the treatment for infection? Because that makes no sense, except we say that about cancer all the time. And when I sit there is that really, no, we have to understand there is no, cancer is a cobbled together clinical state made up of very distinct diseases, all right? And finding one, researching HeLa cells in culture for all world will never lead to the clinical treatment, let's say, of acute promyelocytic leukemia with all transretinoic acid and arsenic, all right? Just like, looking at the behavior of E. coli is not going to give you the treatment for HIV, even though they're both infections. All right? One thing that does tie together all cancer is perhaps that this is really the presence of acquired genomic alterations in our somatic cells. All right. If one way you can think that perhaps infection is someone else's nucleic acids in bacteria, viruses, parasites that make us sick. Cancer is really acquired changes in the subset of our cells in the genome that at least have normal proliferation makes us sick. Even though there are exceptions to both these in both infection and cancer, this is a reasonable way to kind of hold this set of diseases together. All right, and this is compared with, for example, inherited disease alterations in the genome that make you sick, like, for example, sickle cell. All right, and the other thing I always think about is, how put this in perspective. Also, how do we diagnose cancer? Right, what's the test for cancer? And really, there is nothing. Right, there is the only thing we have is you get a lump. Someone takes a piece of it. Someone takes a piece of it, looks at it in the microscope, and says, "This isn't right. This doesn't look like normal tissue." And there's really, it's the diagnosis of cancer from a visual examination of tissue histology by an experienced pathologist. It's a pattern recognition tool. And in fact, when you have rare cancers, you have a unusual sarcoma or a brain tumor, you send it off to Chris Fletcher at the Mass General, and he looks at it and tells us it's sarcoma, crunchy sarcoma class 5. And always the question is, how does he know? All right. And it's kind of like sending a painting of unknown provenance to an art dealer. All right, and getting kind of an estimate. And similarly, you send it to several different experts, you might get several different descriptions of what the disease may be, especially in rare cancers. All right? And our classification of cancer is still, today is primarily, dominated by anatomy and histology. All right, if you even look at the labels of cancer, we have breast cancer, lung cancers, where does the lump arise? And then how does it look like under the microscope? So if you have a lump that primarily arises in the lung, all right, that looks suspicious. We take a piece of look at it in the microscope, and the pathologist says, "Hey, this isn't right. This is a cancer." And then literally, if the cells are small, we call it small cell cancer. If the cells are not small, it's non-small cell lung cancer. And then looking at the patterns of morphologic growth under histology is then further subdivided into adenocarcinoma, squamous cell carcinoma, or other histologies. All right, and this is kind of our current, still a basic histological big subtype. But this kind of classification of cancer masks an enormous underlying heterogeneity of what the actual molecular etiology is. So for example, if one looks at adenocarcinoma of the lung and looks at what are the molecular events, what are the genomic alterations in that individual cancer that's driving its growth, you see there's an enormous heterogeneity. All right, and this is really important because, for example, 4%, let's say, of these non-small cell lung cancers will have alterations in a gene called ALK, or anaplastic lymphoma kinases, the rearrangements, all right? And those cancers and those alone will respond beautifully to treatment with drugs that target the ALK kinase. And so this begs the question, are ALK inhibitors lung cancer drugs? Well, 96% of lung cancers don't respond, all right? It's really an ALK pathway drug, and it treats a set of diseases that are addicted to ALK signaling. All right, and similarly, there are a small percent that have HER2 mutations, BRAF alterations, et cetera. And so then, in some ways, we don't know how to treat lung cancer, right? We do know how to treat EGFR mutant non-small cell lung cancer. We know how to treat ALK altered lung cancer. And so it's clear that this molecular etiology is going to be a very important addition to our anatomy and histological based classification. And in many ways, this again parallels, my wife was an infectious disease doctor. And so, oncology is only 50 to 100 years behind ID. And in the old days, infectious disease was also classified by anatomy and histology. We had pneumonia clinics and encephalitis clinics. And imagine when you organize, which is not a reasonable principle when one understands etiology. And I always used to joke that, if you treat pneumonia with penicillin, you get a 30% response rate, let's say, depending on where you are and the local epidemiology. But I would say that oncology will look at that and says, you're only getting a 30% response rate because you're not giving high enough doses of penicillin. And we should give really high doses of penicillin until the patient seizes and see if you can improve response rates. And in some ways, that's what we've done in cancer. All right. And we got the expected results from that kind of strategy. So the other important advance that occurred over the past 10 years is that our ability to rapidly identify the set of genomic alterations in individual cancers has really dramatically expanded. So for a long time, to sequence a part of the patient's genome or even part of the genome required, intensive labor, months of labor, and lots of money. But now, with the advent of next generation sequencing, which is really massively parallel sequencing, this has really solved sequencing and, more importantly, unlocked clinical samples, because almost every patient with cancer has a formalin-fixed, paraffin-embedded piece of tissue somewhere. And the problem with formalin fixation and paraffin embedding is that it preserves the morphology really well, but it fragments DNA and introduces random base pair changes because of formaldehyde crosslink. Right? And this made standard sequencing very difficult to do in these clinical pathology samples. However, next generation sequencing, or really massively parallel sequencing, overcame those obstacles, because the way next generation sequencing works is that you take your DNA and you fragment it. So little small fragments that are found in formalin fixed tissue are totally fine. And the other thing is that the way it's done, you get multiple reads across bits of the genome, so that random base pair errors that are introduced to any one given bit of DNA by formalin cross-linking can be averaged out. All right, and so this really allowed sequencing to work in our standard pathological samples. This was initially found by Levi Garrett at the Farber and has really changed the ability to do these kind of analyses in standard tissue. And right now, basically, genomic sequencing is solved. So you can take your favorite bit of the genome, all right, like your favorite megabase of the genome, have it sequenced to high depth and get the data back from a clinical sample in about 7 to 10 days. And in fact, the time from these labs getting the formalin fixed tissue to generating sequence data is less than 48 hours. The rest of the time is really the analysis of the sequencing data and the generation of the report. So now the stumbling block is no longer even sequencing, it's the analysis of the sequencing data. All right. There are challenges associated with sequencing clinical specimens, and that is, the big challenges are basically purity. Often in a clinical biopsy specimen, the cancer cells only represent a minority of the nuclei in the sample, especially in a diagnostic biology. All right, in a diagnostic biopsy. And similarly, there's also heterogeneity. Cells may be made of multiple subclones, which may be the source of future resistance. And because of this, you have to have something called a robust measurement of allele frequency. All right, or to put it in perspective, let's say you're biopsying a lung cancer, all right, and the sample purity is 20%. So 20% of the DNA is coming from tumor cells. The rest is coming from normal cells in that biopsy. All right, and let's say you're looking at a heterozygous mutation, say in KRAS. All right. All right, so that's present in one of the 2 DNA molecules of KRAS in the tumor. So that means overall in the sample, 1 out of every DNA molecules that cover KRAS will have the mutation of interest. All right, so you have to be able to detect robustly that 10% allele frequency, 10% of the DNA molecules will only have that mutation present. And early on with our old PCR sequencing methods, it was difficult to figure out what is there because, with very tiny amounts of DNA, one of the wild type alleles could win the PCR race, all right, if you weren't careful, and you get lots of false negatives. But the cool thing is that with hybrid capture, current modern approaches, you can get now coverages. You can get independent reads of more than 500 depth. So you get 500 different DNA molecules that are read. And so with that, you can really robustly identify low mutant allele frequencies, because often the sample purity in some of our biopsies are actually way less than 20% and down to 5 or 10%. And we can still, with this approach, really identify those calls. And importantly, we also get rid of false negatives. All right. And there we have to go a little bit into technical details of how sequencing is done. You imagine that if I, lay down PCR primers, sequence up a bit of DNA and put it on an next-gen sequencer and sequence it to 10,000x, it looks like I've got great coverage, but I don't know when I start with a little bit of DNA, if I use PCR, whether those 10,000 reads that I got off the sequencing machine, do they represent just amplification of one DNA molecule for my sample, or do they really represent 10,000 different DNA molecules? All right, and that's really important when we have these kind of low purity samples. The way hybrid capture-based sequencing works is that we take DNA first and we fragment it, and fragmentation is random. Then you add your PCR primers here shown in red at the end. Then you amplify, then select for your parts of the genome of interest. And the cool thing here is that every read will have a different start site. And if there's a PCR, if there's a lot of reads that come from amplification of one of these reads, you'll see multiple reads that have the exact same start site, and those are PCR duplicates, and you can toss it. And so when you get depth in hybrid capture-based next-generation sequencing, we say, we sequenced this region to a depth of 470. That means we've really looked at 470 different DNA molecules in that sample. And that's really important because let's say I have a patient with melanoma and I'm looking for BRAF mutation, which I have really good drugs for. If I do PCR sequencing, it's negative. I don't know really if it's negative or not. However, if I do hybrid capture-based sequencing and I've sequenced 500 different DNA molecules and none of them have the BRAF mutation, I have a very good negative predictive value. And so I can withhold that therapy with confidence. All right. Based on this, about now, over 8 or 9 years ago, we started a protocol for targeted genomic analysis of human cancer at the Cancer Institute, which basically patients with rare poor prognosis cancer were identified. They were consented. Samples of both peripheral blood were taken, and already existing FFP tumor blocks were retrieved from pathology. And then we did sequencing of a panel of about 300 genes. Initially, this was in collaboration with what was then a startup, Foundation Medicine, back then, and now it's agnostic. There's a whole bunch of platforms that work just as well as our original collaborator. And then these data are reviewed at a molecular tumor board that meets every week. And then data and recommendations tend to see coalition, and then clinical data on treatment and outcome are gathered. Patients are forward consented, so we can also get excess tissue and do research sequencing analysis, prepare explants, xenografts, cell lines as needed. And the key thing is these aren't hardly identified, so if these become important, we could take these data through an honest broker, validate them through a clear certified protocol, and represent them at our tumor board. And patients are forward consented, so if they have recurrence progression, we can rebiopsy and do this process multiple times during their history. And so initially, we want to see, does this actually make us do anything different than our standard approaches? And so we looked at our first 100 patients, which enrolled relatively rapidly, and found that in our first 100 patients, when we did this, we could get successful high quality sequencing on already existing samples in more than 90% of these cases. And most of these specimens had alterations in the set of genes we looked at, and that we could actually, the tumor board actually recommended clinical action or trial in a majority of these patients. And then we asked, okay, this is what the tumor board recommends. How often was this therapy that was recommended actually implemented? We found it was implemented about a third of the time. All right, and the reason it was implemented in part was because we have a very robust suite of early phase clinical trials through our phase 1 program at the Cancer Institute. However, the majority of patients, no treatment was implemented even when there was something potential. And part of that was either there was no accessible trial or drug. So, cancer doesn't travel very easily. And the other thing we also noted was that even when there was a trial available or an appropriate treatment, often patients were getting sequenced, after 4th-line therapy, and their clinical status deteriorated even before a trial stock could open up, and they, went on to hospice, and realized we need to sequence earlier and often during the course of treatment. And so this is our first, 100 patients. We now have, several thousand patients through this protocol, and our data have been very illuminating. To give an idea of kind of some of our early kind of experience with this, I can give you a case. There was, which is one of our very early cases, which was a 64-year-old gentleman with recurrent metastatic papillary kidney cancers, which is an unusual subset of a renal cell carcinoma. And the patient was treated with multiple agents, including standard agents at that time, as well was sent to a clinical trial of a drug called ARQ-197, which was supposed to be a potent inhibitor of oncogene called MET, because it was known that a subset of these papillary renal cell carcinomas actually have MET alterations. But he had rapid progression even on that treatment. So then he came back to us and we entered this protocol and we sequenced him. And to our surprise, he had a classic, he had a mutation in MET that ends up being a classic activating mutation in the kinase domain. And in fact, in vitro data suggests that this would be highly sensitive to certain already present inhibitors, including this drug called crizotinib, which is an old inhibitor that can inhibit both the ALK kinase as well as MET kinase. All right. And we were surprised because he'd already progressed supposedly on a MET inhibitor. All right. And so we treated him with crizotinib, and he actually had a dramatic response. So this is his CT scan before treatment. And then 2 months after, as you can see, nice resolution of the pleural effusion, as well as these pulmonary nodules, as well as improvement in his liver lesions. And in fact, his major symptom actually was intractable hiccups, which really affected his quality of life from the pleural effusion. And these cleared almost within a week of starting therapy. And this is informative. Unfortunately, this lasted only about 4 to 5 months before the tumor started progressing. But this is important because it also helped us identify that ARQ-197 was not really a MET inhibitor. Because here's a patient with a classic MET inhibitor that progressed on this treatment. And then when we treated him with a well-known reversible MET inhibitor, he had a nice response. And in fact, data came out after this that ARQ-197 didn't really function as a MET inhibitor. So that's like, this is a simple case of, here's a target treatment that we treat with. And over the past now 7-8 years, we've used the tumors, this kind of protocol as really as an engine to drive both basic and clinical research. We've used this to investigate the molecular etiology of rare cancers. We looked at, we enroll a lot of our patients in our early phase trials on this to look at the mechanisms of exceptional response to both immunotherapy as well as targeted therapy, and learn a lot about how to interpret and use the current approaches of genomic analysis. Now, today I'm going to talk to you a little bit about, to contrast that original story, something that a patient that we treated about 3-4 years ago that really shows how far we've come in our ability to analyze genomic data, as well as the challenges, the many challenges we still have. And so this is a case of a adolescent with metastatic colon cancer, which is a very rare situation. I was actually on the clinical service, on the adult service, and I was the pediatric service consulted us. And it's always not good when the adult oncologists are consulted on a pediatric case. And so this is a girl presented with abdominal pain and found to have a large colonic mass and multiple liver lesions. And an obstruction, was treated with a diverting ileostomy, and then tumor was biopsied, and this showed to be an adenocarcinoma of the colon. There's no family history. She was enrolled in our protocol, and the sequencing showed that there was a mutation in a gene called BRAF, as well as mutations in PTEN, SMAD, and P53. Right? And this is a classic kind of composition of a subset of colorectal carcinomas. The patient was started and treated with standard chemotherapy, which is FOLFOX. And she initially had a good response. So she had an informative tumor marker called CA19-9, and it was up at about 1400 when she was diagnosed. When we initiated palliative FOLFOX chemotherapy, she had a nice reduction in her tumor markers and improvement in some of her liver metastases. But as we know, this lasts about 3 months, and then she started having clinical radiographic and serologic progression. And so now the question is, what do we do next? We know usually second-line therapy is then given as FOLFIRI, and usually second-line chemotherapy lasts about half as long as first-line chemotherapy. And then after that, third-line chemotherapy, and et cetera, in this disease, is not very effective. So the question is, hey, can we action the mutation, BRCA6, which is in this patient? And BRCA6 is one of the early mutations in the signaling pathway that was identified initially in melanoma. And you guys to remember, BRAF is downstream of the signaling pathway down from receptor tyrosine kinase. Here, for example, EGFR, you have RAS activation, and BRAF is downstream of RAS. So when RAS is turned on, it binds to and dimerizes BRAF and brings it to the cell membrane. And then BRAF is a serotonin kinase that's activated and then starts a cascade of downstream kinases, MEK and ERK, that then lead to a stimulation of cancer cell growth. And early on, work by a variety of investigators, including Keith Flaherty, noticed that if you inhibit either BRAF with an inhibitor called vemurafenib, or you inhibit downstream with a MEK inhibitor, you can get dramatic responses in melanoma. And so, for example, this is single agent BRAF inhibitor causing an impressive change in progression-free survival when compared with standard chemotherapy in patients with advanced melanoma, shown here in the right. But intriguingly, even though these inhibitors are extremely active in melanoma, they were not active in colon cancer harboring the same mutation. So unlike in melanoma, where the BRAF inhibitors, either alone or in combination with MEK inhibitors, cause a 70% response rate, the response rate in colon cancers harboring the same alteration was 5%. And so the question is, why are colon cancers with this mutation insensitive to this treatment, whereas melanomas are quite sensitive? They harbor the same mutation. All right. And so this is where kind of basic science comes in. There was a very lovely study from Renee Bernard's, and also Jeff Engelman's lab at the Mass General also found similar data at the same time, which tried to address this question. So why are BRAF mutant colon cancers not responsive to BRAF inhibition, whereas BRAF mutant melanomas are? And so here, for example, are cell lines. In blue is a melanoma cell line, and this shows the response to increasing doses of this BRAF inhibitor, which ultimately is vemurafenib. You can see the blue line is exquisitely sensitive to increasing doses of the BRAF inhibitor. However, these are a set of colon cancer cell lines, and these not so much. Their sensitivity is marginal at best to these inhibitors. And so Bernard's and his group at the Netherlands Cancer Institute asked the question, okay, they had the hypothesis, maybe these guys are insensitive to vemurafenib because there's another kinase signaling pathway that can step in and promote survival. And so they asked the question, hey, if we knock down every kinase, will knockdown of any of these kinases through shRNA, will knockdown of these kinases now make the BRAF inhibitor lethal in these BRAF mutant colon cancer cells? So this is a kind of a subtraction synthetic lethal screen using shRNAs. We won't go into details of that, but the cool thing was the major hit when they came out of this was the EGFR, epidermal growth factor receptor. So if you knock down the epidermal growth factor receptor in these colon cancer cells, now all of a sudden they're much more sensitive to BRAF inhibition. And that's really interesting because we use epidermal growth factor receptors, including antibodies against EGFR, in the treatment of colon cancer. And we know actually the marker of resistance is mutations in RAS or BRAF. Those that are actually resistant to EGFR inhibitors given us by themselves. All right. And so in fact, they went on to confirm the results of their screen. So this is a colony formation assay here. And you can see here that these are colon cancer BRAF mutant cells. And as expected, they're insensitive to treatment with either an EGFR antibody or a small molecule inhibitor of the EGFR kinase. And they're relatively insensitive to treatment with the BRAF inhibitor. But now all of a sudden you combine them. And now all of a sudden the cells are exquisitely sensitive to the combination. And this is interesting because it's not how we normally combine drugs in oncology, right? So we like to see single agent activity and combine them in combination regimens. And here are two drugs which are known, have no single agent efficacy, but when combined, all of a sudden may be effective, at least in vitro. And the reason for this is really interesting. And I'm going to walk you through this only set of Western blots I'm going to show. And the idea is, if you remember, downstream of BRAF is the kinase MEK, which should be phosphorylated, and the kinase ERK, which we can see. And we can see a baseline in these BRAF mutant colon cancer cell lines. There is enormous phosphorylation of MEK at baseline. Here you see by the phospho-MEK signal, as well as ERK. When we add on the BRAF inhibitor, the phosphorylation of MEK goes away and ERK goes away. So the drug is doing its job. It's on target. It's inhibiting BRAF. But all of a sudden, at the same time, when the patient drug it, now all of a sudden EGFR gets phosphorylated and turned on, and other signaling turns on. Phosphorylated AKT, a side pathway, also gets turned on. Now, when you inhibit both EGFR and BRAF, none of the signaling pathways are able to turn on. You inhibit everything, and the cells die. All right, and this is the concept. This finding led to the concept of what we call adaptive resistance in colon cancer, in which when you have in colon cancer, which are very EGFR dependent, when there's strong downstream signaling, there's actually a feedback inhibition mechanism where ERK actually comes back and inhibits EGFR activation. Now, when you disinhibit, when you inhibit BRAF, you release this inhibition, and now EGFR can now signal through alternate methods, including the PI3 kinase pathway, and support proliferation. And so now we have to hit both this and this in order to get cancer cell death in these BRAF mutant cancer cell lines. And in fact, they're able to show in xenograft models that that even though EGFR inhibitor alone and BRAF inhibitor alone were not effective, in the xenograft models of BRAF mutant colon cancer, the combination is quite effective. All right, suggesting the context of the mutational profile matters. And what's nice is that this came out in Nature, and then within a few years, this was translated into phase 1 studies. And here's a phase 1 study showing a combination of a MEK inhibitor, a BRAF inhibitor, plus an EGFR inhibitor. You can't read the numbers here, but so I say this is actually a very active regimen, even at lower doses. And actually, this past year just got FDA approval for some of these combinations for the treatment. Right? But in our patient, what about us? When we're treating this a while back, there was no data in pediatric patients, even though we had a trial going on here. And so this is, learned us that we need to really reduce the age of entry for our investigator-initiated trials here to 12, as that's supported by the NCI, so we don't miss these patients. So what do you do with our patients? So after, kind of knowing the natural history of disease, meeting with the leaders of speaking with the leaders of these early phase trials, and with the patient's assent and her parents' consent, we initiated therapy with triple target therapy with an EGFR inhibitor, panitumumab, BRAF inhibitor, dorfafenib, and trametinib. And we saw a very nice response, radiographically, serologically, and clinically. All right. But of course, this didn't last. So we put on initiated therapy, and the therapy worked for about 4 to 5 months, and then she started having progression. All right. At this time, this is back in 2016, we also started to monitor her with a new what was new technology at that time was looking at circulating cell-free DNA. And I'm going to describe this a little bit. So the idea is that cells in our body are dying, and when they die, they release free DNA into circulation. And cells can die from apoptosis, where you get these small, uniformly sized DNA fragments that wrap around about the size of nucleosomes, and necrosis, which gives larger, unequal sized DNA fragments. So in fact, if you look at free DNA in plasma and do a sizing column, you get a biphasic peak, a small peak from a smaller size peak from apoptosis, and a larger, broad smear from cells which have died from necrosis. The problem is, of course, is that you have to be very careful in how you actually look at cell-free DNA to make sure that the DNA you're not getting is just contamination from lysed white blood cells. So the analytic capabilities of capturing cell-free DNA are very critical, and now have been solved. And the cool thing about cell-free DNA, it has a very short half-life. So there's mostly enzymatic digestion of unprotected DNA in our serum, and has a half-life of about minutes to hours. There also may be hepatic clearance, and there is some evidence of maybe renal clearance. This is important because there's really renal clearance. Theoretically, you could detect this circulating DNA in urine. And there are some companies and studies looking at that. All right. And the idea is that, imagine in your body, you have both healthy cells are dying and tumor cells are dying, and both are releasing fragmented DNA into circulation. If we could capture this DNA and sequence it, you imagine that the relative ratio, we can not only identify the presence of mutations that may be coming from your cancer, let's say the BRAF mutation, right? But the relative ratio of DNA that is mutant to DNA that's wild type for your gene of interest may actually tell you something about the tumor burden. The more tumor you have, the greater the fraction of DNA that's circulating that's going to be mutant or carry the mutation of interest. All right, so this may be kind of like a universal tumor marker. All right, and pretty story short, back then we started following her with a serial analysis of ctDNA, and the way these data are presented is that these presented in these fish plots, and the width of each of these different colors, each of these different colors represents a different mutation that's present that's seen in cell-free DNA, and the width of these bars represents the relative allele frequency. And you can see that when she was responding, she had very little detectable circulating DNA. At the nadir of her response serologically, we couldn't detect any mutant DNA in her blood. And then when she's progressing, we can all of a sudden detect reasonable quantities of mutant DNA in her blood. And what's intriguing is that now she's developed resistance. Theoretically, if these DNA is released from cells, and the cell has a BRAF mutation here, shown in dark blue, p53 mutation here in light blue, and SMAD in green, they should be in relative ratio of 1 to 1 to 1. But in fact, you can see there's there's greater allele frequency of BRAF than there is of these others. And this actually suggests that there's BRAF amplification. And in fact, we look carefully at DNA without going into details. There was evidence of both BRAF amplification and MET amplification of a gene called MET in this at the time of her resistance to targeted therapy. And this is important. Initially, these were the early days of cell-free DNA, and so we didn't necessarily believe that we can get amplification calls from looking at peripheral blood. So in fact, biopsied one of her liver tumors that grew on targeted therapy and compared it to her biopsy at diagnosis. You can see that the biopsy resistance, in fact, had evidence of both BRAF amplification and MET amplification, as we see. So this is concordant with what we're seeing in just looking at cell-free DNA from her blood. All right. And so now the question is, okay, she's been treated, she has resistance, and she has evidence of amplification of MET and potentially amplification of BRAF. What do we do next? Well, interestingly, there was just at that time we're getting this data, there was just data from the Nicolanto group in Italy that in fact MET amplification is a known resistance of to BRAF and EGFR blockade in BRAF mutated colon cancer, and they may actually respond to MET inhibitors. Drugs like crizotinib, that same drug we treated that patient with papillary cancer. Unfortunately, this patient was progressing quite rapidly, and it still takes us about 2 to 4 weeks to get a drug which has no FDA approval in pediatrics for treatment. And so we said, this is an unusual situation where here's a patient with BRAF mutant colon cancer who's been off chemotherapy for about 6 months. And so we said, why don't we initiate second-line chemotherapy while trying to get this a drug? And what happened then was that she actually had a very nice, so we initiated FOLFIRI, and she had a very nice response. All right, I've seen here serologically, radiographically, and also by cell-free DNA, she went to undetectable in several months. All right. Unfortunately, this did not last. So the response lasted for about 3 to 4 months. It's almost as long as the first chemotherapy response, and then she started progressing. All right, seen here by her tumor markers. But this is where we continue to follow her by serial ctDNA, and this is where the data becomes surprising, is that here's her tumor come back. We can see that as her tumor is starting to progress, we can see the cell-free DNA levels start being detectable in blood. But here you can see that when the tumor is progressing here, there's something intriguing. Here you can see that the BRAF allele frequency here, this dark blue, is much greater than the light blue and the green. But here all of a sudden it's normalized, it's 1 to 1 to 1. And in fact, the clone that is growing under chemotherapy did not have evidence of the resistance mechanisms that were seen in the when the tumor was growing under targeted therapy. So there was no evidence of the MET amplification and no evidence of BRAF amplification. So this strongly suggests that, hey, these cells that were selected as being the resistant clone under pure targeted therapy were not the same clones that were now rising on resistance to chemotherapy. And in fact, that these may be non-cross resistant. So the cells here, all right, don't show the molecular features of resistance to targeted therapy. The cells here that arose under resistance to chemotherapy, all right, suggesting these are truly non-cross resistant. So this raises the possibility, hey, if this is true, can we retreat with the same target therapy? And this is something we never do in oncology because we assume that resistance is cumulative and irreversible. So we go from regimen A to regimen B to regimen C. We never go back to regimen A. All right. And in fact, she actually had a mixed response. On irinotecan, certain liver lesions were actually gone and others were progressing. And so there were just at that time, data came out of phase 1 trials showing that you could safely combine irinotecan together with vemurafenib, BRAF inhibitor, and cetuximab, an EGFR inhibitor. And so what we did was we maintained the irinotecan of FOLFIRI and just added back on a BRAF inhibitor and an EGFR inhibitor. And she had another dramatic response. All right, showing that this clone that's arising on resistant to chemotherapy still, even though it's resistant to chemotherapy, was now sensitive again to targeted therapy. All right, so it was actually you're getting different clonal selection under different selection pressures, and it shows that chemotherapy and target therapy are truly non-cross resistant. And and we can see the same response in ctDNA analysis too going on. Right, and to give a long story short, we were able to do this several times in her course, and so now we're over 2 years from initiation of her treatment, and suggesting that this was dynamic cancer treatment, not just based on start regimen A, then switch to regimen B based on statistics, but really identifying the mechanism of resistance and then treating, and then also knowing when these mechanisms kind of arise and actually go away under different selection pressures. And this really tells us that, hey, that the chemo and target therapy are truly non-cross resistant. And in some ways, we did the stupidest approach here, where we treated with one approach, waited until gross resistance, and then switched. And you imagine that we should have done combined or intermittent therapy up front. And in fact, the other interesting thing we noticed was that if you look at the composition of clones in different metastases, they had very different, not all metastases had all resistant clones. So there were some metastatic lesions, so here's a large liver lesion that was present at diagnosis that went away with FOLFOX, initial chemotherapy, and never came back. Right? There were others that came up on resistance to initial chemotherapy, went away with targeted therapy, but then grew back, and then went away with chemotherapy and then grew back. Kind of this had probably a mixture of clones, or was vacillating with different therapies. And the cool thing is that even metastases that occur late, so here's a metastasis that arose late in her therapy, all right, on resistance to FOLFIRI, this went away when we reinitiated target therapy, and again, this did not come back. This never progressed again, showing that different metastases may actually have different mixtures of these clones, and raises the idea that, hey, we should consider focal treatment in some settings for particularly for some resistant areas, other areas of responding. All right. And to kind of follow up on this, because this is an interesting anecdote, we want to always convert what we see in the clinic to both basic and clinical research studies, is that we work with Mike Verzi's group at Rutgers, who actually developed a model of BRAF mutant colon cancer. He found a very interesting thing that in order for cancers develop in the mouse intestine driven by BRAF, you have to also knock out SMAD, the differentiation factor. And so similar to our patient, these tumors have both BRAF mutations as well as SMAD mutations. And we were able to show that organoids developed from these tumors, this year it's work done by Kevin Tong and Mike Verzi's lab, all right, are resistant to both BRAF inhibition alone and EGFR inhibition alone, but are sensitive to the combination. So this model works just like the human cancer. And interestingly, they're also sensitive to standard chemotherapy. And so we're using these organoid-based models to figure out, what are the right ways to combine targeted therapy and chemotherapy upfront or alternating to provide data for a development of future clinical trials. This kind of data where we see kind of when we're now using targeted therapeutics, where we can and have ability to monitor development of molecular resistance, we see this kind of pattern of clones that come, resistant clones that come and then vanish, when therapy is changed or removed multiple times. And I'll give you just one or two more quick examples of this to show how this works. So again, this is a patient with now melanoma who's BRAF resistant immune, all right? And this patient, this is a CT scan. This shows a large axillary lymph node here. And when treated with single agent vermorafenib, this is the old days, the patient had a very nice response that lasted about 4 months or so, and then started to progress, okay? Actually, interesting, some liver lesions actually went away and also started progressing here. But when we, what we did was we looked at we biopsied the tumor before treatment, sent that for sequencing, and then we had a biopsy of the tumor after progression, and we sequenced that. And at baseline, the patient had a BRAF V600E mutation as well as these other alterations. And then on progression, what was different was now there was a presence of a novel fusion, rearrangement in the BRAF gene that put it in frame with a gene called AGAP, which is nearby. And so this suggested that this tumor that had both of these alterations is now resistant to single-agent vermorafenib. The cool thing is nowadays we can see, we characterize this fusion that was present in this patient's tumor, showed what the fusion looked like. And nowadays we can just synthesize this fusion, introduce it into cells, BRAF mutant cells, and see what happens. So we're able to show that if you have these parental BRAF mutant melanoma cell lines, they're normally very sensitive to BRAF inhibition inhibitor. But if we now introduce this fusion into those same cells, now all of a sudden you induce marked resistance to single agent vemurafenib. However, these cells remain sensitive to downstream inhibition by MEK. And this is just the Western blot showing the signaling pathways there. Right? And in fact, we treated the patient with a MEK inhibitor and they had a nice response. But then the story gets, took a turn that was unexpected to us at that time, which was, the patient actually at that time, we added trametinib and a MEK inhibitor. The patient initially responded, but then the immunotherapy trials came along. And the patient was appropriately interested in getting on the new immunotherapy trials that were coming on back in 2014 for melanoma. And so there was a trial going on here. So we stopped all the treatment with the inhibitors and then started treatment on the PD-1 antibody early phase clinical trial. Okay. Unfortunately, he had initial pain going on this, and ultimately he ended up having no response. His tumor just progressed through this. Because the mass was growing in the early days, we decided to give him a chance and actually resect this axillary mass. And this was taken. So this is the same site that was biopsied before and shown to have the BRAF fusion resistance. And when we looked at the sequencing results from the axillary mass on progression on immune checkpoint therapy and off of all targeted therapy, you can. So this is the tumor at pre-treatment. This is on resistance to vemurafenib when I had this AGAP BRAF fusion. And now when it's progressing on immunotherapy, there is also, we don't see evidence of this AGAP BRAF fusion anymore. And we know it's not a false negative because the intronic region where this junction was found was sequenced to a depth of over 200. So it was present at least 1 in 100 cells in that specimen. We should be able to detect it, and we were unable to see it. So what happened? And so one can imagine that maybe the immunotherapy selectively killed these cells and let everything else grow, or what we think may be happening is that this clone that had both the BRAF sensitivity and the BRAF fusion was actually only more fit than the parental cells during therapy, when in the setting of ongoing BRAF inhibitor therapy. So it's kind of like antibiotic resistance, where the multiple antibiotic resistant bugs are actually less fit than the sensitive cells. They're only more fit when antibiotics are present. All right. And actually, we tested that theory by taking, again, our cell lines that are either parental or BRAF mutant or have this AGAP BRAF fusion, and mix them and show that if you kind of put them, we labeled so we can see one is green and one is ungreen, and we can make mixed cultures. And what we found is that if you put them together, the resistant cell line actually, if you, in the absence of any inhibitor, it doesn't grow faster. In fact, it grows more poorly than the parental cells. But now, if you treat the cells with the BRAF inhibitor, now the ratio is reversed. And so what's happening is that you mentioned pre-treatment. Actually, these guys are there is rare cells that have this BRAF fusion. They're actually less fit. They can't outcompete the wild type cell, the ones without the fusion. In the setting of vemurafenib treatment, now these guys dominate. But now when you remove the inhibitor, these guys no longer have the advantage over the parental non-mutant cells, and these guys dominate again. And we're able to show that in vitro also in this patient. And this kind of can be seen kind of over and over again. I won't take you through this case just for instance. Again, in this case, there's a patient with a lung cancer initially present with activated mutation in EGFR as well as a P3 mutation. They were treated with a first-generation inhibitor, erlotinib, and they did well for a year. On progression, they developed EGFR amplification and MET amplification. Interesting, similar, kind of similar themes that come up, for example, in our patient with the BRAF mutation. MET amplification is a driver of resistance. So here, back in 2013, we added crizotinib onto erlotinib, and the patient did well for about 2 years. But when they progressed, we biopsied the progressing lesion in the lung, and now all of a sudden they had EGFR L855R, and now they developed another mutation in CIS that prevents erlotinib binding, and the MET amplification was lost. And in fact, this is a different clone because this clone no longer had a P3 mutation and instead had CDKN2A2 loss. So this is a clone that's related, ancestrally related to this clone, but actually an older clone that now was selected under this combination treatment. So interestingly, in 2016, osimertinib, which is the third generation EGFR inhibitor that actually tackles the T790 mutation, was available, and we treated with that. And the patient did well for a year. And then when they progressed, we biopsied the progressing nodules. And what came back? This clone. This clone now is no longer had T797 mutation, and is in fact now it's back to this meta-amplification EGFR amplified P3 mutant clone that came back. And so now we're treated with osimertinib plus crizotinib, and the patient responded for a while. So now here we are, over 7 years out from diagnosis. And instead of, in the old days, we give erlotinib, they progress, we just give chemo and then nothing. Here, because we're able to see what the mechanisms of resistance are and then put on treatment, we were able to get continued responses for a while. And we wonder, if up front with this patient been treated with osimertinib plus crizotinib, maybe we could have really had long-term remission in this patient. All right. The last thing we'll talk about, just a brief case. We also use our ability to analyze molecular to really investigate, exceptional responders in our early phase clinical trials. And I'll give you one quick example of that. Is there's a patient with endometrial cancer who had, was chemo resistant and had bulky adenopathy in the periodic retroperitoneal lymph nodes. And she was enrolled in a phase 1 trial of PD-1 antibody, pembrolizumab actually. And she had, so this is her bulky lymphadenopathy pretreatment. And within 8 weeks, you can see this one region, resolved tremendously. Her lower extremity edema resolved. And actually, she has been in this progressed to a complete response, and she's been in remission for over the past 5 years, and now 3 years off treatment. All right. And so the question is, why did this patient have this exceptional response to pembrolizumab? Whereas most patients with endometrial cancer or most cancers do not enjoy this kind of response. And so we sequenced her. She was a 283rd patient sequenced in our protocol. And we sequenced her from a lymph node biopsy back in 2014. And what we found was that she has like a large set of mutations. There were 32 mutations and 52 genes. Usually when we do this kind of report, we see like 4 or 5 mutations, these enormous list of mutations. And so this is an enormously high mutation burden cancer. And why is that? Well, interestingly, there's a mutation in DNA polymerase epsilon, or POLE, a hotspot mutation. And to guys remind you, DNA polymerase epsilon is the leading strand polymerase of DNA synthesis. All right. And so during S phase, this is the leading strand DNA polymerase involved in DNA synthesis in our cells. And part of its job is it not only synthesizes DNA, but it has a little bit of proofreading function. So if it extends and puts a base on and it actually makes a mistake, which it does, occasionally, it actually recognizes that mismatch. It backs up, cleaves it with an exonuclease as the proofreading function, and then goes back. So it has its own little spell check function as it progresses. These mutations, like the V411, disable this proofreading. So the polymerase functions but can't proofread, and so it causes a high error rate. All right, and so this causes tumors that have enormous mutation burden. And this is important because there's principles are coming along then that tumors that have high mutation burdens may be immunogenic, in that even though this tumor has lots of mutations, most of which don't affect cell fitness, the presence of these point mutations may lead to new peptides with protein amino acid changes that lead to epitopes that weren't present during immune maturation and now may be detected as foreign antigens by the cell. So these are point mutations in proteins that don't affect cell fitness, but may be immunogenic. And maybe this is the reason why these tumors respond to immune checkpoint therapy, because in order to survive, they have to induce local immunosuppression. Right? And in fact, people have shown that other tumors with high mutation burden, like those with mismatch repair, are also quite sensitive. But this patient was microsatellite stable, but had high mutation due to a different mechanism, this polymutant. And then we were able to show, we did an analysis, bioinformatic analysis, saying if you look at all patients that have this kind of hotspot POLE mutation and compare them with microsatellite unstable tumors or microsatellite stable tumors, not only do these tumors have an enormous mutation burden, but they have clear evidence of immune activation, high expression of the immune checkpoint genes, as well as evidence of a T cell lymphocytic infiltrate. And this suggested that POLE mutant cancers mark another set of cancers that are microsatellite stable, but have a high mutation burden that may be exquisitely sensitive to immune checkpoint therapy. And in fact, this has led to us a basket trial for this in looking at other repair defective cancers that's ongoing at our Cancer Institute as well as other sites looking at polymutants, other DNA repair mutations, to see if these presence of that will respond to single agent pembrolizumab, and has led to basic science investigations as a collaboration between my laboratory, the laboratory of Eileen White, and Ned Latimer, in which we are using these polymutants to develop mouse models of cancer that have high mutation burden, because there's a real lack of mouse models that have an intact immune system and can respond to immune checkpoint therapy. So we're manipulating POLE to create tumors with high mutation burdens and see if these mouse tumors are now responsive to immune checkpoint therapy, and give a model for us to figure out how to rationally combine immune checkpoint therapy with other tumors. And so with that, I'm going to stop this and say that ultimately, the technology, what's really cool is that technologies and abilities to do molecular analysis of cancers that before I could only do in the laboratory or in models can now be routinely done in clinical samples from patients. And I think one of the major problems we have in cancer is that I don't think we've classified our diseases optimally. All right, and the real problem is, we want to classify diseases. We want to, imagine you have a real disease and you have a treatment, you should have like, a 90% response rate. If you have a 30% response rate, you have a collection of stuff. And the question really is, how do you optimally cancers by phenotype, genotype, epigenotype, and ultimately, how do we reimagine the basic workflow of pathologic-based diagnosis? Right now, our entire system is aimed to preserve the histology and morphology at the expense of the molecular information. Formalin kills DNA and RNA. All right, and the question is, how do we reimagine this so we preserve both these pieces of information and don't sacrifice the molecular data and capture that up front? And that way we have these data up front, and how do we do that, with minimum distress, expense, and time? And imagine that in the old days, what we had was we had pathology in which we had a histologic pathology based on anatomy and histology, and then we treat. And then what happens if, for example, you treat non-small cell lung cancer with EGFR inhibitors, only 5 or 10% will respond, and it's really hard to detect that in clinical trials. Imagine now what we're doing is we're looking at genomic pathology in addition to histologic pathology, and we can classify all the tumors that have alteration-specific genes, and perhaps put those in clinical trials and now see strong responses in smaller numbers of patients enrolled. And this is kind of, led to a new kind of way we do clinical investigation in oncology. And the idea ultimately is that in addition to our traditional classification, we'll also get molecular information that will really allow us to guide therapy that targets not statistical, 20% respond, but attack the underlying molecular etiology of individual cancers. And that this is causing us some problems, because now instead of just lung cancer or breast cancer, within breast cancer, there's probably 30 different cancers, and we have to figure out how to classify those and do clinical trials individually for this large collection of rare cancers that our diseases are fragmenting into. And of course, we have to think about other aspects as opposed to just purely DNA sequencing. And the idea is now, we think nowadays about, lung cancer, and, we have to think about a certain fraction of KRAS, certain fraction of EGFR mutations, et cetera, and some have BRAF mutations. Maybe we really should be thinking about, no, there are BRAFomas. There are cancers that are driven by activation of BRAF, and they can arise in different cells of origin. And to optimally target that, we may have to use different combinations and different cell lines, as you saw in our patient. But maybe this is a better classification, because this gives us information, okay, on how to treat. And ultimately, that's why we classify stuff, all right, is to give us information more than the name. With that, I'll stop. I'd like to acknowledge, obviously, I presented work from members of my lab, our collaborators at CINJ, including the Systems Biology Group, our collaborators at Rutgers and at other institutions, as well as industry and our funding sources here. That I'll stop. Happy to take questions.
Thank you so much. That was fantastic. I think it was a wonderful example of what can be done in the lab that translates directly into improvement in our patients' care and their lives. So thank you. That was fantastic. While we're waiting, I know we're already at the top of the hour.
Sure.
I guess you started to touch on this at the very end in your second to last slide, but I was curious. I think that case that you brought up of the patient with BRAF V600E mutant colon cancer that didn't respond to the agent that's typically used for and effective in melanoma. I'm wondering about the effect of the local environment. And you in your second to last slide, there was a mention about the microbiome, but what other kind of epigenetic influences outside of just the mutation might impact its behavior in different.
So colon cancer is a great example of that, where in fact is a signaling, there is a local signaling network, all right, the EGFR dependent signaling network that really need to be in it. So BRAF clearly is driving the growth of that tumor. That is the oncogene, but to target it effectively in that cell of origin, we have to use a little a different approach. And then on top of that, ultimately, I think these genomic drivers are causing the cancer, but obviously there's going to be, both, differences in the host and other things that might impinge upon this. The microbiome is really interesting in some ways because the host interactions are particularly important in our immunotherapy approaches, which I didn't discuss much here today. And those may play a particular role when we're marshaling not just cell intrinsic features to target therapy, but host features to target the cancer as well.
Wonderful. Great. Thanks so much. I know we're already past 9 o'clock. Laura Willett just said, thank you. It's a brand new world in oncology. It sure
I
is.
apologize for going over time. This is
Thank you. Thank you so much. Thank you everybody for attending.
all right.
Great day. Thank you so much.
All right. Thanks so much.